Prepare to experience the strongest radio allowable by law. Secrets will be revealed. Myths dispelled. From the studio gym where excuses never apply, it's Superhuman Radio with your host, Carl Lenore.
Hey, welcome back to another episode of Superhuman Radio. Today is Monday, November 16th, 2020. For those of you listening to this show 100 years from now and realizing that this audience knew a lot more than anybody else did, we have a fascinating show today. We're going to discuss the microbiome during the first hour. You know, that's a favorite of ours to talk about. And then later in the show, we're going to talk about a method of determining whether or not rheumatoid arthritis drugs will actually work therapeutically for people who suffer from RA. There's a way to figure that out before you even get started in a therapy, which is also very interesting. Before we do any of that, we have to thank our title sponsor, Legendary Foods. EatLegendary.com is the website. SHR10 is the code. Not only do you save 10% off everything you buy there, and everything is great, but you also let them know you heard about them here on Superhuman Radio. So use the coupon code often. And don't forget to pick up a box of the new Hot Fudge Sundae Tasty Pastries. For those of you who are new to this, the Tasty Pastry is basically a Pop-Tart that has had all the sugar removed, is gluten-free, and has high quality, high protein, high leucine protein in it. In the case of the Hot Fudge Sundae, it has 15 grams of protein, 0 sugar, and I think it's got like 2 or 3 impact carbs. It's got a little bit of fiber in it. They taste better than real Pop-Tarts. The kids will love them. You'll love them. They can't keep them in stock. So please go to eatlegendary.com, use the coupon code SHR10, and save 10% off, and enjoy yourself. Enjoy yourself. And without further delay, I'll bring my guest on now. And he is Dr. Kirk Bergstrom. How you doing, Kirk?
I'm doing great.
Thanks for being here. So for those of you who were looking at the show and going, well, where's Dr. Lijun Shaw? He had problems connecting. So Dr. Bergstrom has jumped in here to help us out. He is also one of the people involved in this recent paper that looks at some new information about the microbiome. So, Kirk, to get started with this discussion, why was this study needed? What did this study springboard off of? What work was done before? And it could be by your group or other colleagues that these questions still needed to be answered.
Sure. So, Carl, the study really was built on a need to really define the relationship between our microbes in the gut and the copious mucus that's produced. And I can really elaborate on that if you want.
Yeah. So when you say the copious mucus, this has become a more interesting topic because the mucosal barrier is implicated in everything from leaky gut syndrome to actually the ability for things to get into the bloodstream that trigger autoimmunity. So yes, please talk about that mucosal barrier.
You bet. So, mucus is not, the most glamorous thing to talk about. It's when people think about it, they think about snot coming from your nose and sickness and gross, sticky, gooey stuff. But it's a really fascinating, utterly fascinating molecule.
It's definitely fascinating to guys, not so much girls.
Yeah. Right? Well, I won't be the girls interested. So I have, some researchers in my lab who want to study, female researchers, and I'm all about empowering women researchers. And so, but mucus is, it's one of the most complex molecules I think you'll ever come across. It's made of amino acids, about 3,000 to 5,000 amino acids long.
So technically, mucus is a protein.
Mucus is a protein. More specifically, it's a glycoprotein.
Oh, so sugar there. Okay.
Yeah, exactly. Oh, it's got tons of sugars on it. So it's 5,000 amino acids long, and it's got this domain called a mucin domain, which are sites of attachments for sugar structures that we call O-glycans. And these O-glycans, they form, they're basically short chains of sugars that form, made up of about 5 to 6 different monosaccharides linked together in all sorts of ways. They could form, up to 100 different structures on the mucin. And so it makes it extremely chemically and biologically diverse molecule. And then on top of that, so that changes its size from a couple hundred kilodaltons to now in the megadalton range.
Right.
So it's extremely large and chemically complex. And then it forms this massive polymer. It's linked together, and this was studies done by an amazing researcher in Sweden, Gunnar Hansen's lab, to really define the polymeric structure of the mucus. And it's now in the 100 megadalton range. Like it's utterly enormous molecule. So the problem with that, it makes it kind of it's just a huge headache to study because of its complexity and its enormity. So it's just as fascinating as it is as much of a headache to study.
Like the difference between studying structures and looking at a house, which is complex.
Yeah.
Now looking at an entire metropolitan area.
You could think of it that way. Yes.
Yeah. Very interesting. Okay. So
yeah,
the
go ahead.
mucosal barrier serves a purpose, right? Is the purpose exclusively to line the stomach and keep the things in the gut from getting directly out into the bloodstream, or are there other purposes for the mucosal barrier?
Sure. So, one thing to maybe clarify is what you mean by the mucosal barrier. So the mucosal barrier is actually a collection of several things. So the mucus is part of that mucosal barrier. Then you have your epithelial barrier as well, which is a single layer of epithelial cells that also is a part of that mucosal barrier. And what they do is limit the microbes from interacting with immune cells in the lamina propria that are poised right there to attack any sort of invader. And so the mucosal barrier, if you're referring just to the mucus, that is what our research is focused on.
Right. No, and the mucosal barrier is held in place by what do they call them? Cilia, or some sort of tissue that actually has like tendrils, and it creates a rebar, if you will, for the mucus to hold onto, right?
Yeah, actually, in the airway, mucus is produced in large amounts in the airway, and certainly the cilia are more pronounced there than they are in the gut. And so that function you just described is definitely reminiscent of what you'd see in the airway, where the cilia and mucus work together to push the capture material, and the cilia push it up into the airway, and that's what you cough up as like sputum.
Okay.
But in the gut, it's a little bit different scenario. So in the gut, the mucus, actually, it's quite remarkable. It's made of one particular mucin called MUC2, mucin 2. We just shorten it for MUC2. And it really has two completely diverse roles that it plays simultaneously. So one, it forms what's called traditionally called the outer layer. It forms a niche for the microbes. So the microbes love to colonize it, and they feast on the glycans and the sugars in the mucus. And we don't know exactly what the mucus is doing to those microbes, but it's certainly, we think, helping to keep them happy. But at the same time, while it's attracting the microbes, it's also forming a barrier to keep them away from the tissues. And so it's kind of a form of social distancing. You can sort of think of it that way. and so it's kind of remarkable that the mucus system, as we're starting to call it now, because that's a pretty accurate term for it, and this was coined by actually the Swedish group I mentioned earlier. the mucus system is it does those two things, totally diametrically opposed things at the same time. It attracts the microbes and keeps them away from the tissue. And so that's kind of our understanding, but like how it does that is still very much an open question, and that's things where our study kind of comes in.
Okay, so talking back about the mucosal barrier just for a second.
Mm-hmm.
Acid doesn't affect it, right? The acidity of the stomach, does it play any role in preservation, in modulation, in affecting the mucosal barrier at all?
Actually, that's a really, really good question, Carl. So the stomach, the mucus is a little, again, different from the rest of the intestine. The mucus system really changes as you go through the GI tract. The stomach, it's much more neutral, meaning that there's a lot less acidic sugars on it. And the reason is because the stomach is so acidic. The pH in the stomach can go down to like pH 1. 1 to 2, so that's extremely acidic. And so what the mucus does in the stomach, it helps to create a buffer between the harsh acidic environment of the lumen and the tissue, and it plays a really, really important role in creating that very steep pH gradient between the stomach and the tissue, which is really over, it's so steep, it's really over maybe 30 or 40 microns. So that's the importance of mucus in the stomach. Yeah, go ahead.
No, go. Don't finish your thought, please.
Yeah, but in the colon, things, the pH increases as you go, through the intestine, and in the colon, in the large intestines, probably around pH 6, roughly. And so, actually, the mucus is much more acidified in the stomach, believe it or not. They have these really interesting monosaccharides called sialic acid, and also these sulfate groups that contribute to this the acidity and negative charge. And so the mucus has a pretty much quite a different role in the colon compared to the stomach.
So does the mucus play a role in keeping microbes from inhabiting areas of the digestive tract that they're not supposed to be? So we have a lot of people talking about small intestinal bacterial overgrowth. And I kind of think that happens to be tied to, as we get older, the acidity drops. Obviously, the pH goes up, and it allows microbes to inhabit areas that they shouldn't. Does the mucosal barrier protect or create an environment that is either beneficial or not beneficial to microbes from taking up space in places that they're not supposed to?
Absolutely. What's really interesting, Carl, is that there are certain parts of the intestine where actually the tissue really likes the bacteria. The proximal colon actually is a place where microbes are routinely interacting with the epithelial cells, even going deep into these epithelial structures called crypts, and like really in close contact with the host tissue and not causing disease. And so that part of the tissue, for reasons we don't completely understand, but seems to be very well adapted to the microbes. So it's totally fine with having this complex community set in such close contact. But the distal colon, though, doesn't have that relationship with the microbes. It appears to want to keep them away. It is not adapted, and that tissue is very susceptible to bacterial-induced inflammation. So you're talking about, the descending colon, the rectum, in people and in mice. We do a lot of work in mice as preclinical models. It's the same thing. That region of tissue is extremely susceptible to microbial-induced inflammation. And so that's where the mucus plays a role in what's called the biogeography of the microbiomes. It keeps them segregated and in the lumen where that's where the tissue is most happy to have them. But in the more proximal regions, the mucus serves other roles. It's probably more feeding the microbes, but not necessarily keeping them away from the tissue, because in the proximal colon, that's where you get the most beneficial effect of the microbes with producing these anti-inflammatory metabolites through fermentation, like short-chain fatty acids, which can have a direct impact on the immune system.
Butyrate.
Yes.
Butyrate. Yeah.
Propionic acid, butyric acid. Yep.
So talk about your study. How was your study designed?
So the study itself was designed. That's a kind of it's a big question. A lot of the platforms that we needed to support our hypothesis were developed in our previous publications, previous studies. So these include the animal models we used to sort of support and prove our hypothesis in vivo, and some of the imaging techniques. And so as we designed the study, we basically relied on three or four major platforms. And so one is genetic manipulation of the mucus system in the mice. And again, we've developed these mice in previous, but we had to actually generate a new model in this study as well. And then some imaging techniques in order to really look at sort of the big picture, what's going on in the whole tissue. We had to develop sort of an imaging technique where we can look at the whole, what's going on in the entire colon. So this is, an 8 to 10 centimeter structure. We want to be able to see what's going on at one time, so we can understand what's happening with the mucus system as it's being produced throughout the entire colon. And so we had to develop a new imaging platform, and then we took advantage of non-antibiotic mouse models and germ-free mice as well. And then we took also with our collaborators in Los Angeles, some world-class data scientists, John Braun and David Casero. We did some of the most cutting-edge microbiomics that we've ever done. And so in order to really understand what the mucus system is actually doing to the microbes. So I'm happy to elaborate on each of these.
Well, I do that because my next question was, what makes the mucus? So it sounds like, do specific microbes make the mucus?
That's a awesome question. So what we discovered in the study is that basically these cells, specialized epithelial cells called goblet cells, these are kind of your mucus factories. Their whole purpose is to, their major purpose, actually some studies have come out, they have different purposes, but their major purpose is to produce the mucus system that's secreted into the lumen that interacts with the microbes. And so, but where our study really, provides clarity on this, on what they do, is the fact that it's actually the mucus in one part of the gut, the more proximal colon, is what's contributing to most of the mucus that you see. And in the distal colon produces a totally different subtype that contributes little to the overall barrier function of the mucus. But it does contribute to a new subtype of mucus that we discovered in the study.
But this is in the colon. What about in the gut itself? What about the stomach? The stomach has mucus lining it as well. Is it the same tissue, the same cells that make the mucus in the stomach?
So in the stomach, they have sort of mucus cells that we don't necessarily call them goblet cells in the stomach. They just call them more mucus cells. But yes, they do the same thing, but they produce a different type of mucin called MUC5AC. And so this is related to MUC2 that you see in the small intestine and the colon, but it's a different molecule.
Okay.
But it does have a similar structure.
So does it provide what's the word I'm looking for? Environment specific protection that you need in the gut because of the high acidity versus downstream where the acidity is lower? Is that the difference?
Or in the stomach, you mean?
Yeah, the stomach to the colon, let's say. What?
Yeah.
Different type.
Gotcha. Yeah. So let's walk through the whole GI tract real quick. Okay. So starting in the stomach, you have this MUC5AC produced by the mucus cells, and it produces this very neutral type of mucus. There's not much acid component, and that forms that sort of buffer zone that contributes to that pH gradient from that low pH to that more physiologic pH that our cells are more used to. Right. And that's the purpose of the mucus really in the stomach is to create that buffer zone between the acidic gastric juices. Now, as you move to the small intestine, now you're getting to the goblet cells. And so the small intestine is quite different from the colon. The small intestine has these structures called villi.
Mm-hmm.
And so these like stick out like fingers, of course, into the lumen. And
Right.
goblet cells are produced along the epithelial part of the villi.
Right.
And so they also produce mucus. And here, the mucus cooperates with the epithelial cells, which secrete antimicrobial molecules that bind to the mucus. And also through Paneth cells in the small intestine, that also these will bind to the mucus and contribute to antimicrobial activities of the mucus. But the mucus doesn't form really a barrier in the small intestine. It forms like this stringy mass that that really isn't much of a barrier at all. And so we think probably the antimicrobial component of that mucus is able to, compensate for that lack of barrier function there. And so, but then you get into the colon, which doesn't have villi, it just has these crypt structures, these invaginations.
Right.
And so that's where you get that really strong, thick barrier structure that it's now sort of a classic structure that you would see in the colon of these mice. And that's where the mucus, its main function there is to promote both. It's supposed to feed the microbes, but also to separate them from the tissue, perform a barrier. So it's really more about the microbes in the colon, and in the stomach, it's more about the acid.
Right. Okay. So do all of these cells that produce the different types of mucus use the same raw materials to make mucus? Like I've heard that if you want to reestablish the mucosal barrier of the stomach, take glutamine.
Interesting.
What do they use to make mucus?
So to make mucus, you're going to need a whole lot of amino acids. You're going to need a whole lot of sugars and a whole lot of enzymes. So if you want to boost your to mucus, you got to so you're going to need of course the gene to be expressed, the MUC2 gene, for example, in the colon. But that's not all. You're going to need enzymes that are going to decorate the mucus with those sugars. So as the mucus is being translated to the protein, and that's getting processed, and then post-translationally processed, moving through the Golgi apparatus. Now you have to have a lot of glycosyl transferases. These are the enzymes that will add the sugars, the unique monosaccharides, to mucin and build on those glycan chains that I was telling you. And so they need to be present and active. And so it's actually, we don't know. What it is as it moves through, the glycan chains get built, and then you get at some point, once it moves through the Golgi, it gets packed into these mucin granules after it's been glycosylated, and then it forms this polymer, and then that polymer within these granules is then released. So really, what you need to understand if you want to boost mucus is you want to understand not just the mucin gene itself, but you need to understand all the glycosyl transferases that the mucin gene normally interacts with to make a normal mucus.
Yeah, because it's fascinating to me because there's emerging evidence that as we age, it could be age, or as we live longer and expose ourselves to things in our diet, in our environment. We even know that RF is affecting microbes in the gut now. This mucosal, the protective barrier of the gut seems to thin, and it leads to things that are in the gut that shouldn't be in the blood getting out of the environment and into the blood, and hence autoimmunity, but also just ulcers and IBS and even some say colon cancer as a result of the inflammatory impetus in areas where you don't want inflammation. And it seems to all come back to this, this dielectric, this protective barrier that can go away as we age. Is there any evidence that there's a way to reestablish these a maybe a dietary, a supplementation, a combination of both, a way to improve the protective barrier that's in our digestive tract?
Again, I really appreciate your questions, Carl. So, one of the thing our study was we found is the mucus production is really controlled by the microbes. So, if we take germ-free mice, what was remarkable is that the mucus system was very rudimentary. There's not much of a barrier at all. And even though these mice are on a high fiber diet, you can see the fibers scraping along the mucosal tissue, and the tissue was perfectly happy. It wasn't damaged. And I was a bit surprised by that. So it's really when you only added back microbes, even just one microbe, now you got the production of this mucus, and you get this, and mainly in the proximal part of the colon, and so the microbes really control the mucus production. So that, I think, gives us a lot of insight into how we could, envision a way to boost it.
Which microbes were implicated in this process?
So we looked at a complex community. So in that case, it's hard to nail down the particular one. But we did a study, an antibiotic study, and the Bacteroides theta omicron was one bacteria that was able to induce it on its own, although a slower dynamics than a complex community. So even a single microbe could do it. Yeah.
I mean, so what you're talking about here could be the holy grail in helping people with disorders like IBS and so on.
That's right.
So we did a show a couple years ago that showed that certain carbohydrates, grains most specifically, seem to cause flare-ups of IBS. And and I wonder sometimes, I keep going back to our diet, right? Because look, you're talking about fiber. Right now, we have fiber fortified foods where we probably shouldn't have fiber. So people are getting a ton of fiber that they may or may not need today. We don't know what this is doing to the gut. In fact, Dr. Leslie Aiello, who used to run the Wenner-Gren Foundation in Manhattan, and I had a few conversations about this, and she said that as we have evolved as humans, we have consumed less and less fiber. Our guts got smaller as a result of that, and our brains got bigger. Now, that's a very oversimplistic progression over millions of years. But the point is that we've gone back to eating fiber like it's, like we're supposed to. And I often start to wonder if the fiber isn't somehow causing some unwanted changes in the microbial diversity of the stomach. So with this Bacteroidetes, what was it called? Bacteroidetes what?
Theta iota omicron, or we just call it B theta for short.
Okay, B theta. So yeah. How does that respond to the soluble and non-soluble fibers? Does it do anything with them?
Well, that particular bacterium is what's called a sort of glycan generalist. It has, I think, for last count, maybe close to 100 different enzymes, or basically groups of genes that can utilize pretty much almost any type of glycan. So it can harvest and act on plant glycans, any sort of glycan monosaccharide and linkage that you see in there, all the different sugars that are in the fibers. But it could also act on the host glycans too, which are quite different from the plant glycans. So not too many bacteria have that same degree of flexibility.
And that shows its importance of hierarchy, it's so adaptive, it can adjust to whatever it's being given to do its job.
That study was published by Jeff Gordon's lab back in 2005, which basically that was one of the more transformative studies for me. And so Justin Sonnenberg, who has his own lab in Stanford and a leading sort of bacteriologist and also glycobiologist as well, he's also a co-author on the study we're talking about today. But that was one of the defining things about that, is that bacteria is able to adapt to its changing environment. If you get rid of the fibers,
evolutionarily, what's the word I'm looking for? Important. If when it's that flexible that it knows it has to do its job. So it's going to use all sorts of fuel forms to be able to do that. We have to take a quick commercial break.
Sure.
When we come back, we're going to get back to what you discovered in this study. We're talking today with Dr. Kirk Bergstrom. He's filling in for Dr. Lijun Shaw, who had difficulty logging on to our video system. We're going to take one quick commercial break. We'll be right back with more Superhuman Radio. Stay tuned.
Thank you.
Everybody wants to feel better, be healthier, and happier. If you don't sweat profusely on a regular basis, you won't feel as good as you could. Studies have shown the power of infrared sauna. Infrared penetrates 1 to 2 inches into the body. It helps those with back pain, fibromyalgia, arthritis, depression, and anxiety. Patients get relief from infrared sauna. The Good Health Sauna has been shown to be an exercise mimetic, conveying many of the same benefits as cardio. There are a lot of reasons people buy a Good Health Sauna, but the bottom line is you will be a much happier, healthier person by using the Good Health Sauna every day. You'll crave it, you'll love it, and it will be the best purchase you have ever made. Good Health Saunas provide commercial-grade saunas for in-home and commercial use, backed by the best lifetime warranty in the industry. Unmatched customer service and the best financing makes owning one easy and will fit any budget. Go to goodhealthsauna.com/superhumanradio to learn more and save 25% off.
When craving a snack, reach for Select Savory Snack Sugar-Free Biltong, the healthy alternative to sugar-laden beef jerky. They use a fattier cut of beef, making it more tender and flavorful. For mealtime, choose from their 63 seasonings, which are sugar and additive free. From chipotle rub to savory pumpkin spice, you'll find an option to suit your palate. Go to shrnetwork.biz/SSS today. Use code SHR15 to save 15% on any meat snack or seasoning. Savor the flavor today. Mind Bullet is a kratom product derived from the coffee family to boost energy and creative juices. Mind Bullet comes in both pill and liquid form for the ultimate pre-workout supplement. When needing a little pick-me-up or to get through the morning fog, Mind Bullet will set your productive course fast. Go to shrnetwork.biz/mindbullet, all one word, today, and save 20%. No subscription required. That's shrnetwork.biz/mindbullet. Get yours today. New Mass Pro Synthogen X2 just upped its own legendary game. To distance itself even further from the rest of the pack, Synthogen X2 now has double the key active ingredients. If you've ever wondered what steroid-like recovery feels like, Synthogen X2 delivers. See why others compare it favorably to powerful bodybuilding drugs at synthogen.com. Mass Pro Synthogen. When you train with it, you'll gain with it. Want to improve mental energy and focus? Try the new Superhuman Stack from Pure Nootropics. Stack Dynamine, Salbutamine, Theocrine, and Palmitoyl ethanolamide together and feel the lights go on in your brain. Find your perfect mix of these four. Carl likes 1 cap of each and 2 caps of Dynamine. Bam! Lights on. Go to shrnetwork.biz/superhuman stack, all one word, today, and get yours for 25% off plus free shipping. That's shrnetwork.biz/superhuman stack.
Whether your goal is to build muscle or burn fat, you'll find everything you need at Redcon One. Need help getting a good night's sleep? Try Fade Out, or the most popular pre-workout supplement on the market today, Total War. Sign up for their new Transformation Challenge and win $10,000, or shop for apparel that people at the gym will know that you are serious about your training. Need a testosterone booster that works? Check out Boomstick. Whatever you need, you'll find the best quality supplements on the market at Redcon One. Go to redcon1.com. That's REDCON, the number 1.com, or go to superhumanradio.net and click the Redcon One banner ad today.
This is the Superhuman Channel, doing reps with the weight of the world.
Welcome back. You're not going to want to miss tomorrow's show when Shannon Penna comes on and gives us 2 different low-carb recipes for turkey stuffing. This is a big frustration of mine for Thanksgiving. I want to eat stuffing too, but I don't want all that carbs. I don't want the bread. I don't want the pro-inflammatory grains in my body. Well, we have 2 recipes, one with a low-carb keto bread that you have to make first, and then the other one using cauliflower. So don't miss this show, because you're going to want this in your bird, figuratively speaking, this year. Getting back to one other thing I wanted to mention, Dr. Bergstrom, is I did a show probably about 6 years ago on how acellular carbohydrates, i.e., flour, anything made into a flour from a grain, corn, acellular carbohydrates feed the wrong gut bacteria in the stomach because they're broken down so small. Typically, you'd have to digest them, and they wouldn't really start to become a problem until later down the journey out of the body. But because they're available right in the stomach for these microbes to eat, it actually changes the diversity of the gut to those that are less energy intensive and more have a higher ability to extrapolate or extract nutrition from food, which leads to obesity. And I thought that was fascinating too, because it's these unwanted microbes get fed by flours, any kind of flour, not almond flour, but most of your starchy flours. So, okay, so what exactly did you learn? I don't think I can hear you. Are you mute?
Oh, there we are.
Good.
Oh, I'm good now.
You don't have to mute. I mute you on this end. So
Cool.
So talk about the study. What really jumped out at the study? What is the thing that's really reshaping our understanding of the gut microbiome that was derived from this study?
I think the biggest thing is just really how our guts handle this vast community. So, I think the thinking before was that, we have this microbes that exist in our gut, and, it's hard to sometimes picture. We maybe have this picture that the microbes are just there, maybe floating around. They're, attached to components of the diet, maybe not super organized. And it's just this continuous thing going on in the gut. And so, we think about the gut, we think about the microbes, and we maybe have this picture, of a mucus, for example, separating them, but we don't really have this, the big picture of what is really, really happening. And so, what our studies showing is that you have this massive community that your mucus in your first part of your colon, first part of your large intestine, so the proximal colon is, I should clarify, the first part of your large intestine is actually your cecum, and then you move to the proximal part of the colon. That mucus in the proximal colon is actually secreted into the lumen, and as it travels down, it somehow, reasons we don't understand completely yet, but it encapsulates that microbial community. So it wraps itself around that entire mass in these sort of mass units, I guess you could call it. And so it forms this barrier that is pretty effective at keep sequestering the microbes all together in the community and keeping them from escaping that community.
So they almost become biofilms, it sounds like we're talking about here, right? It creates almost like a biofilm, because a lot of biofilms are designed to sequester unwanted things in the digestive tract, right?
I think a biofilm, I wouldn't necessarily call it biofilm. I would basically, when you think of your fecal mass, that will have probably some biofilms within it. You may have little micro colonies and things, attached to, some of the fibers that are also going to be encapsulated in by this mucus.
Okay.
But it's basically taking that whole community and kind of helping to wrap it all up into a distinct sort of mass. And then that basically community doesn't seem to be able to escape that. I mean, it's not absolute. You're always going to see situations where you see some microbes escape, but in the end, it's really...
it becomes like a virus vault, if you will, on your computer. Like, for some reason, the body doesn't want those microbes to come in contact with the tissue in that area.
Exactly.
So it sequesters it. Okay.
Exactly.
So when that process goes awry, then do we see the types of anomalies to the colon, IBS and all these other, and even colon cancer? Do we see those being triggered by the failure of the mucus to sequester those microbes?
I would answer yes to that, and I could totally base that on elaborate on that. So in animal studies, absolutely. Once you mess up that ability of the mucus to encapsulate, that microbes now are able to have unrestricted access to the tissues as that fecal mass moves through the colon, and that unrestricted access will lead to chronic
Inflammation.
Chronic inflammation. It will hyperactivate inflammatory pathways, and just keep them on an always on sort of situation, leading to this chronic inflammation. And that inflammation itself creates a pro-tumorigenic environment, and which can transform normal cells into tumor cells, and it just basically from there it's just really creates a perfect storm, and then you get the tumors and the cancer, and that's actually a study we published back in 2016.
Wow.
So yeah.
No, go.
So what our study does really, it really points to reinterpretation though of a lot of the previous studies, including our own, where we're understanding what's happening to the microbes, and as you see them, that sort of that host microbe interaction dynamic. Our study really helps to now turn it from like this continuous idea that we're kind of used to seeing to actually it's a very discrete communities that are moving through the colon at one time, thanks to the mucus largely. And so it creates these masses of communities, and in between them it's like these microbe-rich communities, and in between these fecal masses that are encapsulated, you have very bacterial pore zones. So it's a much more discrete communities that you see, rather than this continuous microbe colonization of the tissue of the mucus along the tissue. So the mucus is actually, the previous model was that we're challenging, is that the mucus is not necessarily produced locally and attached to the tissue. It's a very highly mobile structure that moves with the microbes.
Could you use radioisotoped glycans to see where the mucus goes? You know what I mean? Kind of tag it, and then you say, "Oh, this started here, and it went here, and then it went here." And then could you do that?
Actually, we did something very similar to that in this study. So we did a metabolic labeling study using a sort of bioorthogonal approach, where we've used this technology by Carolyn Bertozzi. She's the one who sort of brought this out into the limelight about over 10 years ago now. But it's using click chemistry, so you can use an azide labeled glycan, and you could inject it into the mouse, and it gets incorporated very easily into the mucus. And then you can afterward, once you collect the tissue from the mice, you can do the chemical reaction right on the tissue, and it could show you exactly where that labeled glycan where it is. And we used it basically to analyze the secretory response. But from that information, we were able to get some insight into where it was actually being produced.
So whenever we talk about the gut, we talk about the microbes, but there are also fungi that live in the gut. And from what I understand from a show that we did years and years ago, I'm going to get these terms right. One is a eukaryocyte, and the other one is something else. One has a nucleus and one doesn't. Microbes don't have a nucleus, but fungi do. So they're actually a step higher organism than microbes. And we also know that fungi use microbes the way we ranch, cattle and sheep and chickens. One fungi can gather 10, 20, 100 different microbes that it uses, because these microbes, when they eat something, they poop out something the fungi likes. So they're like little microbe ranchers, if you will. When you study the microbial diversity and the effects of the gut microbiome, do you look at the fungi and what they are contributing to a lot of this stuff as well?
I think you should. This study really focused on the bacteria,
right?
the bacterial component of the microbiome, but certainly, our study has ramifications for all the other aspects of the microbes, including the fungi and the viruses and the eukaryotic sort of microorganisms, the single-celled animals that you may see in there, even some of the worms. All components of the microbiome are going to be wrapped up in this mucus, not just the bacteria. So it's interesting to see what's happening to those components as well. And what was interesting in our study was that even though we discovered, the microbes induce this mucus and their own essential encapsulation, the mucus itself, its sugars, also modulated the microbiome. So you have this sort of counterpart to that. And so it shifted the community, and it influenced their metabolic output. And so we focus on the microbes, but it'd be really interesting to see what this encapsulation and its effect on the community is happening to the other members of the microbiota, as you mentioned, like the fungi.
Yeah, because I kind of feel like looking at the gut and strictly looking at these microbes, but not also looking at the contribution to all these other things that you just listed. We could be making assumptions, and we could be really far off without taking into account how they attenuate and modulate the effects of these microbes and what they put out.
Sure, absolutely. And that's where you could take advantage of things like the metagenomic data that we generated in the study. So we did a shotgun metagenomics, and so now we have basically all that information, at least on the genetic level. See what types of the fungal species that are there, the viruses, and even potentially some of the functional potential that they have by looking at the genes that the fungi would have in there as well. So we could still go deeper in the study. I mean, and use some of the data we generated to actually address those very questions.
We're going to take our last commercial break for this interview and wrap up this discussion. I have a couple more questions I want to try to get covered. Then later in the show, we're going to be talking about a new way to determine whether or not certain drug treatments for RA will actually work for you. They can actually now help identify you as a candidate for some of these potential therapies. So stay tuned. You're listening to and watching Superhuman Radio. We'll be right back.
All right.
Do you remember those delicious toaster pastries you had when you were a kid? The rectangular sugar-filled snacks? Well, guess what? Legendary Foods has just made low-carb toaster pastry. This is the first of its kind, and honestly, these things are amazing. They have 3 to 4 net carb, less than 1 gram of sugar, and 9 grams of protein. You can eat them right out of the wrapper or lightly toast them. The only question is, which flavor, strawberry or brown sugar cinnamon? They're available at eatlegendary.com and Amazon.
Quest Nutrition makes bars, cookies, chips, and pizzas out of complete dairy-based proteins. Our products minimize net carbs and sugar without sacrificing taste. Each delicious chocolate-flavored chip, cookie chunk, and crunchy crumble is custom-made to maintain Quest macros. It's time to enjoy foods that work for you, not against you. It's time to enjoy your Quest.
Proper hydration can lead to improved endurance, performance, and recovery. When we sweat, we lose valuable electrolytes that must be replaced. That's why Element created a zero-sugar, science-backed ratio of electrolytes that you can add to your water. It comes in 3 flavors. Sample them all risk-free with the Element Variety Pack. Carl's favorite is raspberry. Free shipping on all U.S. orders. Go to shrnetwork.biz/LMNT and get yours today.
I love beef. And if you love beef, listen up. I've discovered the best tasting beef in the world, and that's not an exaggeration, at Piedmontese.com. The Piedmontese breed is famous from Italy for being lean and unbelievably tender, with half the fat and calories of traditional beef. Even typically tough cuts are tender when it comes from the Piedmontese cows. And for the first time ever, Piedmontese cows are being raised here in the USA. Get 25% off all beef and free shipping on orders of $100 or more at Piedmontese.com with code SHR. Go to PIEDMONTESE.com and use code SHR today. You will never eat any other type of beef ever again.
Want to improve mental energy and focus? Try the new Superhuman Stack from Pure Nootropics. Stack Dynamins, L-Butamin, Theocrine, and palmitoyl ethanolamide together and feel the lights go on in your brain. Find your perfect mix of these 4. Carl likes 1 cap of each and 2 caps of Dynamin. Bam! Lights on. Go to shrnetwork.biz/superhuman stack, all one word, today, and get yours for 25% off plus free shipping. That's shrnetwork.biz/superhuman stack. New Mass Pro Synthogen X2 just upped its own legendary game. To distance itself even further from the rest of the pack, Synthogen X2 now has double the key active ingredients. If you've ever wondered what steroid-like recovery feels like, Synthogen X2 delivers. See why others compare it favorably to powerful bodybuilding drugs at synthogen.com. Mass Pro Synthogen. When you train with it, you'll gain with it.
You're listening to the Superhuman Channel. We're ripped and we're ready.
Welcome back to Superhuman Radio. We're talking with Dr. Kirk Bergstrom, and we're talking about your microbiome. You know, the microbiome is kind of like outer space. You know, we think we know a lot about it. We don't know a whole lot about it. We're learning all the time. So, was, other than the key findings, was there anything that jumped out at you that you didn't expect when you did this study?
You know, one of the things that I mentioned before was just that really intimate association of the mucus response to the microbes and not to other components of the diet. I mean, we only really tested this with one sort of high fiber diet, so we can't say that other diets, you know, wouldn't have an impact on the mucus, for example, in a germ-free system. But that was quite striking to me. It really goes against this idea that, you know, the mucus barrier is just, you know, the barrier effect is passive to the a passive consequence of the need for lubrication as the stool moves through the colon. But really, the microbes controlling it indicates it's more than that. It's more of a barrier and that niche that I was talking about earlier in the study. So that was really interesting. The other is how the mucus is apparently constructed. So the model before was that it seems to be, it was constructed locally in the tissue by the goblet cells. It sits on the surface, and then it's linked to the surface, and then it moves and gradually produces that niche layer from the barrier. Our study is showing that it seems to be constructed more in the lumen itself once it's secreted. And so how that barrier is actually forming is still an open question. So that was really surprising. And then maybe how the microbes responded as well. So we mentioned that the B theta earlier. What was surprising is that that microbe, when we lose the glycans, that microbe actually went up in terms of its relative abundance. I would thought because it really likes the host glycans, it would probably go down, or at least, but it actually seemed to improve, and so that was a bit surprising. So,
well, doesn't that sound like a reactionary? Like if the mucosal barrier is thinning, the body goes, we need to make more, so it establishes more of those B theta microbes, perhaps.
You know, that's very true. And one way to test that hypothesis would be to, remove both the host glycans as we do genetically in the mice, and also remove the dietary glycans. Maybe give them a diet that is devoid of these complex dietary plant polysaccharides, more simple sugars, and see how the bacteria responds there. And that will tell us whether that ability to improve when the host glycans are lost are actually it's a adaptive response of the microbe in that situation. But I guess it was just surprising because it kind of gives an idea that even maybe the host glycans are actually, limiting somehow, maybe, I don't know, directly or maybe indirectly by its effect on other species, the ability of that particular microbe to thrive in a normal mucus environment. So that to me was actually quite surprising.
So diet has the greatest. So when I first started to study the microbiome for shows,
mm-hmm.
8 to 10 years ago, we started talking about it. It was just fascinating because I thought, oh, especially when you look at things like fecal transplant, the dramatic effect of a fecal transplant. I mean, some of the stories are astonishing. And you go, oh, so you just have to change your microbes to change your health. But as I've gone down this path, I've learned that the microbes are more of a remnant of your diet. And we've done shows that show that microbiome diversity can change in as little as 48 hours just by changing the diet. And we see this in people who go to extreme elimination diets, and especially in response to emerging autoimmunity. We know that people who give up a lot of plant-based foods and go keto or carnivore or ultra low carb, they see almost a remission of a lot of their symptoms, and over time they seem to get better. And this is not an N=1 thing. We have millions and millions of people in this country that if someone took the time to set up a website and get their stories, we'd probably have some really statistically significant information out there that would lead to other studies. So with that being said, the diet changes the microbiome, and the longer you alter the diet, the deeper those changes go and take. Have you found anything that seems to be contraindicated or that seems to disrupt the development of this needed mucosal barrier in various places in the gut, but most importantly in the colon?
Well, we haven't really looked at that specifically, to be honest. But I know other researchers have. I know Justin Sonnenberg, for example, has shown if you're to change the diet, you can impact the microbiome over generations. You can lose particular members, and some of these changes can be compounded as the mice propagate and they transfer their microbes to their offspring.
You're saying that those changes are transgenerational.
Those are transgenerational. Yes, yeah. But those are
the mom gives a lot of the microbes to the offspring, so that makes sense.
Yeah. But the diet will impact that, and then the combination of the diet and then the transfer of the microbes over time will compound those changes originally introduced by the diet.
Interesting.
Yeah, it was very, very, very interesting, actually.
The problem with dietary interventions for issues related to microbial diversity is that some of these microbes, and we remember these are evolutionarily adapted species of microbes. And some of them will become spores. They don't die. They just go into animation. And as soon as you start eating the crap food again, all your problems return. So you can't, it appears to me that once the microbial diversity of the gut has been changed for the worse, you can't diet your way into fixing it, but you can diet your way around symptoms, if that makes any sense.
Yeah, I think I would agree with that. So certainly some of the species you mentioned, the spore-forming like Clostridium species, that's a dynamic of the microbiome that's still really poorly understood.
Right.
And you can get at that through, I think, the evolution of microbiomics, which is getting now into the metatranscriptomics versus just the metagenome. So instead of just looking at what genes are present, you're looking at what genes are actually activated within a bacteria. And so this is Curtis Huttenhauer has been huge at this, and so John Braun, our collaborator in the study, also has been on this. So that's certainly one way to get at it, because you can see if the species is present but not transcriptionally active, that may indicate they're dead, or maybe they're in some sort of a spore state or some inactive state. And so I think it's a totally different dynamic of the microbiome that we need to understand.
Last question.
Sure.
Is there a way to test for the accumulation of the mucosal? I don't even want to call it a barrier, because it's more of a player in the colon that seems to be the culprit that could be leading to chronic inflammation. Is there a way to like ultrasound and go, oh my God, you've got none of that mucosal lining, we need to do something about that?
One of the most exciting outcomes, I think, of this study is that the mucus system that we are normally used to looking inside the tissue actually is attached to the fecal mass. And so you can now analyze the mucus now just looking at the feces. Yeah. You get a lot of insights. And so this answers really addresses a lot of past problems with the mucus, in that now you can get access to it in non-invasive way. So you don't have to, this reduces the number of mice maybe you need to use if you want to look at the impact of the mucus, but you also get access to the mucus through the fecal material now. You can extract it. We've shown in the study, you can extract it the same way as you can from tissues and do glycomics on it and things like this, just from the fecal matter itself. And so what this does is open doors for clinicians, for example, and patients who are, people are interested in the mucus, what's happening to it. For example, if you have inflammatory bowel disease like ulcerative colitis, you can look at the fecal material and you can see what's happening to the mucus just by looking at the feces. And so this opens up the door for not only non-invasive sort of analysis of the mucus, but also biomarker analysis too.
Very fascinating. Very fascinating.
Yeah. Mm-hmm.
This is exciting. Look, Kirk, I want to thank you for jumping in. It was unfortunate that we couldn't get Dr. Lijun Shaw on the show because of technical difficulties, but I think you've covered everything fantastic. And I will. Awesome. I'm inviting you back as you learn more.
Awesome. Thank you.
Just email Elisa. We'll get you right back on the air, because this is really important stuff when we talk about the gut, because I am convinced that just about all diseases of modernity start in the gut, and understanding what makes the gut thrive and what makes the gut sick is probably going to lead to a lot of other diseases being eliminated in the population. So thank you so much for being here today.
Can I say one more thing,
Carl?
Of course. So I just want to really acknowledge Lijun Shaw. So he's the study was driven by Oklahoma Medical Research Foundation. That's where I did my postdoc, and that's where it started. And Lijun is an absolute excellent mentor, has tremendous instincts for science. And so the study was driven by myself and Lijun. And so I just want to give a shout out to him, and also co-lead author Dylan Shan, who did a tremendous amount of work as well on the study. Very talented postdoc there. So I just want to make sure that just acknowledge them. They're important people.
Excellent. Yeah. All right, we're going to take a quick commercial break. When we come back, we're going to switch up. We're going to talk about rheumatoid arthritis, which is a big problem in today's environment. And so stay tuned. We'll be right back with more Superhuman Radio.
Thank you.
Thank you. If there is one mineral you should be worried about not getting enough of, it's magnesium. Magnesium is the body's master mineral, powering over 300 critical reactions, including detoxification, fat metabolism, energy, and even digestion is influenced by the presence of magnesium. That's why I'm so excited to tell you about a new magnesium product I've been taking now called Magnesium Breakthrough. It's the ultimate magnesium supplement, easily the best I've ever seen or experienced. With all 7 forms of magnesium, I was able to arrange an amazing deal for my audience, and I guarantee it's the best deal available on this product. Seriously, with volume discounts combined with our custom 10% coupon code SHR10, you can save up to 40% off select packages of Magnesium Breakthrough. And I promise that this deal is only available on this specific website, magbreakthrough.com/SHR. You won't find that deal on Amazon or even on the company's own website. Go to magbreakthrough.com/SHR and use code SHR10 to save up to 40% off select packages of Magnesium Breakthrough.
When craving a snack, reach for Select Savory Snack Sugar-Free Biltong, the healthy alternative to sugar-laden beef jerky. They use a fattier cut of beef, making it more tender and flavorful. For mealtime, choose from their 63 seasonings, which are sugar and additive-free. From chipotle rub to savory pumpkin spice, you'll find an option to suit your palate. Go to shrnetwork.biz/SSS today. Use code SHR15 to save 15% on any meat snack or seasoning. Savor the flavor today. Mind Bullet is a kratom product derived from the coffee family to boost energy and creative juices. Mind Bullet comes in both pill and liquid form for the ultimate pre-workout supplement. When needing a little pick-me-up or to get through the morning fog, Mind Bullet will set your productive course fast. Go to shrnetwork.biz/mindbullet, all one word, today, and save 20%. No subscription required. That's shrnetwork.biz/mindbullet. Get yours today.
Whether your goal is to build muscle or burn fat, you'll find everything you need at Redcon One. Need help getting a good night's sleep? Try Fade Out, or the most popular pre-workout supplement on the market today, Total War. Sign up for their new Transformation Challenge and win $10,000, or shop for apparel that people at the gym will know that you are serious about your training. Need a testosterone booster that works? Check out Boomstick. Whatever you need, you'll find the best quality supplements on the market at Redcon One. Go to redcon1.com. That's REDCON, the number 1.com, or go to superhumanradio.net and click the Redcon One banner ad today. Everybody wants to feel better, be healthier and happier. If you don't sweat profusely on a regular basis, you won't feel as good as you could. Studies have shown the power of infrared sauna. Infrared penetrates 1 to 2 inches into the body. It helps those with back pain, fibromyalgia, arthritis, depression, and anxiety. Patients get relief from infrared sauna. The Good Health Sauna has been shown to be an exercise mimetic, conveying many of the same benefits as cardio. There are a lot of reasons people buy a Good Health Sauna, but the bottom line is you will be a much happier, healthier person by using the Good Health Sauna every day. You'll crave it, you'll love it, and it will be the best purchase you have ever made. Good Health Saunas provide commercial-grade saunas for in-home and commercial use, backed by the best lifetime warranty in the industry. Unmatched customer service and the best financing makes owning one easy and will fit any budget. Go to goodhealthsauna.com/superhumanradio to learn more and save 25% off. Chronic stress leaves us tired, distracted, and physically vulnerable. Apollo Neuro is here to help you take control. Apollo Neuro is a new wearable that trains your nervous system to be more resilient to stress. Apollo's gentle vibrations use your sense of touch to help you recover from stress, going from fight or flight to rest and digest so you can relax, get to sleep, focus, and stay healthy. Developed by physicians and neuroscientists, Apollo Neuro has been tested in multiple clinical trials and is proven to improve heart rate variability, the key biometric of stress. Special offer for friends of Superhuman Radio. Try Apollo Neuro today and get 15% off your purchase at apolloneuro.com/superhuman. That's APOLLONEURO.com/superhuman.
This is the Superhuman Channel, doing reps with the weight of the world.
Welcome back to Superhuman Radio. We're going to be talking about a new study that shows that there may be a way to predict whether or not a particular treatment will be beneficial for those who suffer from rheumatoid arthritis. We're talking today with Dr. Jesmond Dalli. Did I pronounce your last name proper?
That's correct. Yes, thank you.
Thank you for being here. So,
Pleasure.
Let's start. Why this study? What was happening before this study that required this study to be done? Do these rheumatoid arthritis remedies not work for a large portion of the sufferers?
Yes. So in many countries, and I'm currently based in the United Kingdom, but this is also true for the United States and many other countries in the world. Patients with rheumatoid arthritis are treated in a somewhat prescriptive manner. That means that there's a procedure that's followed. That it's not necessarily based on whether you're likely to respond to a drug, but more because the drug is usually cheaper to access, and so physicians are somewhat obliged to provide the cheaper drug first. See what happens. If you respond, then that's all very well and good.
Yeah,
however,
in the United States, we call that the standard of care.
Correct.
The standard.
That's correct. And if you don't, then you've suffered for additional months. Potentially, you've been exposed to some of the side effects of some of these drugs. As we all know, nothing is 100% safe in the world. And so this also means that your disease would have progressed to potentially an even worse state before you might be able to be assigned the drug that works for you.
Well, and on top of that, what happens quite often in this type of an approach where we're going to try this drug. If that doesn't work, we're going to try that drug. when your body is when you walk into the doctor's office, you're sick. You have an ailment you want treatment for, and you manifest certain unwanted, undesirable symptoms, and then they put you on a drug that doesn't work but gives you a host of new symptoms, and now you don't even know if the next drug is just getting rid of it's just a cessation of what you stopped taking, or the next drug is actually working. And it's very sad because patients who don't respond quickly to the first therapy could end up chasing new symptoms that were a result of this merry-go-round of drugs that they use.
Correct. That's very, very correct. And so this is an unfortunate situation that we find ourselves in, because we don't fully understand how many of the diseases occur, what makes these diseases occur, and also how affecting some of the pathways that the drugs tend to target will influence the disease progression in different patients. And in some cases, because we don't understand, because each patient is different, and most people don't only have, say, rheumatoid arthritis. Some people may have diabetes on top of it. Some people may have cardiovascular disease. Some people may have some weight issues, obesity-linked issues. So you know, us as scientists studying something in a laboratory where everything can be made in an ideal case scenario,
Right.
And then going to the real world when on top of everything we have stress, as we are all very familiar with during these times. All these aspects tend to influence our ability to respond to a specific drug. And to date, it's been our ability to predict whether someone is going to respond or not, or actually to evaluate very early on. You know, we're not talking about a magic ball here. It's really trying to understand what the mechanisms of the disease that is of interest, in this particular case, arthritis, are involved, and how these particular mechanisms are regulated differently in different people. We've not necessarily quite got to that level of granularity, and this is, I think, what has hindered us in understanding and being able to tell whether one patient is more likely to respond to the drug that they're being prescribed or not. And so this is the aspect that we've been very keen on addressing over the very many years.
Okay.
And
I'm sorry.
Please.
Go ahead. Sorry, I'm sorry.
And so you know, my laboratory and my mentor's laboratory, where I trained at Harvard Medical School and the Brigham and Women's Hospital, there's a level of pioneering of new biology where we're looking at how the body regulates the immune system, and the immune system regulates itself in both diseases like rheumatoid arthritis, where you have what is generally referred to as a sterile inflammatory response, but also in infections. And we know that these checks and balances in many of these conditions become lost, or at least they don't work as effectively. And so we wanted to base this potential of understanding whether a person is going to respond to a drug that impinges on these pathways, as we also found out, based on how well these pathways are expressed, these particular molecules are produced by the body.
Okay. So how was the study designed?
So the study design was based on, first of all, methodologies that were pioneered in my mentor's laboratory, and then we refined using mass spectrometry to measure molecules that we discovered in the peripheral blood, so in blood of patients that were diagnosed with rheumatoid arthritis, but were not given any of these disease-modifying anti-rheumatic drugs. So this is at the very early stage, and
Right.
we were very lucky to have a fabulous collaborator in Professor Costantino Pizzalis, who had, through no little effort, managed to establish a patient cohort where we could obtain blood from these patients before they went onto their standard of care.
Right.
Exactly, which is very difficult, as you can appreciate, to obtain, and this allowed us to really be able to test this hypothesis to a large level. And then, apart from using these methodologies in this unique cohort, we also layered on top of it an expanding body of literature that is based around artificial intelligence and machine learning. So the ability of a computer-based system, if you will, to churn through chunks and chunks of data to identify patterns that are reliably reproducible.
And what was the molecule that you discovered?
So this is a class of molecules that are called the specialized pro-resolving mediators. And these are molecules that are primarily produced from omega-3 fatty acids. You know, the fatty acids that our parents always told us, you got to eat your fish, you got to eat your fish. And this is one of the reasons why you got to eat your fish, because these fatty acids, these essential fatty acids, are the precursors for the production of molecules, the specialized pro-resolving mediators, that are important for our body to regulate inflammation.
So let's talk about that for a second, because it's a very important distinction what we're talking about here. NSAIDs, corticosteroids, they stop inflammation, but they stop healing, because inflammation is telling the body, hey, come fix me over here. And so when we talk about inflammation, there's two things that you can do. You can halt inflammation, which isn't really a good thing because you can't halt it forever, or you can help it resolve. And by resolving, we mean it goes through the complete process of injury, resolution, healed, all better. These molecules found in omega-3s actually and the truth of the matter is, people who take large doses of omega-3s have been also shown to be effective as well. But these molecules don't just stop inflammation, they speed up the healing process, correct?
That's very correct. I couldn't have expressed this better myself. So biology of these molecules.
How is it that these people who are suffering from this chronic inflammatory disorder are producing their own SPMs from their diet? Is it actually helping them, or do they just need larger doses?
So this is an aspect that actually we're currently actively investigating, and it's a fantastic question, because the disruption and what leads to chronic inflammation, in this case, for example, rheumatoid arthritis, could be from two aspects: the inability of your body to produce these molecules early on enough to stop the inflammation from becoming uncontrolled. And then once you can imagine this as a train, right? If you pull the brakes at an early time, you can stop the train from going out of control. But if it gathers enough speed, then the brakes just don't work anymore. And even though your body produces these SPMs, the system has become so disrupted that you need a reset, a strong reset button. Now, the other side that could actually be disrupted is not just the ability to produce them, but these molecules require proteins on the surface of cells known as receptors that can sense their presence and then convey the biology that they're meant to convey to stop inflammation. So if the expression of these receptors happens to be out of tune, out of sync with the production, or there are defects in either the way the receptor and the signaling that it should elicit downstream, or the conformation of the receptor is incorrect, then that defeats the production in a way, because you have the molecule, but you don't have what is required for it to be conveyed into a biological action. So there are two aspects here that could become disruptive.
Actually, I'm going to submit a third one.
Okay.
Maybe you could put my name in the next study. So,
I will.
so here's the third one. I've done shows on rheumatoid arthritis now for pretty close to a decade. And we know that rheumatoid arthritis has a autoimmune component to it. And we know that autoimmune diseases, by and large, are a result of things in the stomach getting out into the bloodstream that look like bad actors. The immune system goes after them, and then they go, oh, it also looks like thyroid tissue, so let's go after the thyroid too, or let's go after the meniscus and the joints and the chondrocyte. And so could it be the third possibility that these receptors are occupied by something that has a greater affinity to them, and so that the the SPMs can't even attach and do their job.
It is also possible. Receptors have multiple functions. There's not one ligand for one receptor, because otherwise biology would, we would require an innumerous number of receptors, and that's not how biology works. So it's likely what's been known for some of the receptors, and many of these receptors we're still really learning the basics of their biology. But for the ones that we've been studying for a little bit longer, we know that one receptor can signal both pro-inflammatory, so molecules that activate the immune system, versus the pro-resolving mediators that dampen the immune system. And the downstream signaling pathways that are elicited are also not just mediated through the ligand that's binding to the receptor, but how the receptor dimerizes, so forms multiple complexes with other receptors. So that is a valid point, and it could actually be also an aspect that requires testing. I would submit that it's potentially more endogenous ligands, so other molecules that are inflammatory that might interfere with the ability of SPMs. But we know that some of the SPMs, for example, regulate bacterial function, and this is been known. For example, some of the SPMs regulate quorum sensing mechanisms in pathogenic bacteria. So there's no reason why it shouldn't be the other way around. And actually, some of the receptors that we're studying are also activated by formulated peptides that are characteristic of bacterial species. Bacteria tend to have all their proteins are formulated, and so there is potential crosstalk too for this.
This is very fascinating to me, because I have so many thoughts running through my head. Like I have attention deficit syndrome. So and then the other thing, as you kind of pointed out, with the multiple ligands activating receptors, is the mosaic of receptors can be selectively. Domains can be selectively activated, and other domains cannot be activated, which throws a whole different curveball into the discussion. Anyway, so so just talk more about your study. So once you discovered the prevalence of these SPMs in the blood, what was the next step?
So the way we conducted the study was in two phases. The first phase was to identify potential biomarkers. So in the first phase, we obtained blood from a group of patients that we knew retrospectively that they had either responded to the drugs being given or not.
Mm-hmm.
And so what we did here was, using these machine learning methodologies, we queried whether there were specific molecules that were prognostic of treatment outcomes. So whether there were molecules that linked with whether a person responded or did not respond to that treatment outcome. And then by in the second phase of the study, we obtained more blood from completely different patients and questioned whether these molecules that we found in peripheral blood were indeed prognostic. So using completely different patients, could we still classify someone as a responder or non-responder based on the concentrations of these molecules in their blood? And we found that we could do it with an accuracy of about 89%.
Wow.
So that means that we could get 9 out of 10 patients in the right category based on the levels of these molecules in peripheral blood. And the other interesting thing that we observed is that we could not only tell whether these molecules were predictive of whether someone was going to respond or not to these drugs, but also the type of disease that they had. So rheumatoid arthritis, and this is work that's been done by our collaborators, is being classified into different pathotypes, so different sort of, if you will, forms of the disease.
Okay.
And this is based on the pathology, the way that the joint looks under a microscope when people take biopsies from the joint. And it's linked with whether the immune system, which part of the immune system in a way becomes disrupted, whether it's the innate part, whether it's the adaptive part, whether it's more of a stromal part. And we could tell from what's happening in the peripheral blood whether a patient was more of one category type and versus another category. And that's important because we know that different types of disease, different flavors of this disease respond better or worse to different types of treatments too. So together it starts to tell us, it's starting to tell us that we can start now to break down RA patients into more of a unique person response and a more of a designed treatment approach, rather than you have arthritis, so you need to go down this route.
Yeah, you're moving towards individualized medicine, which is actually the goal of medicine today.
Correct.
So the first question that comes to my mind is, there is actually companies out there that sell SPMs. There's a company that has a patent here in the United States called Metagenics.
Correct.
I actually take SPM. They have a product called SPM Active. It's a distilled portion of omega-3s that is very rich in the SPM form. So why not just give people SPMs as a because we know that omega-3s don't just act as an analgesic, but they actually turn genes on and off that help shut down inflammation while resolving inflammation at the same time. Why not just give people SPMs instead of these other drugs?
Well, that's where we're aiming to be. So the product that you mentioned really helps feed the production of SPMs, of pro-resolving mediators, and it does so because so these pro-resolving mediators are produced via the conversion through enzymes of substrate into bioactive molecules.
Okay.
And the advantage of that product is, and I'm not, I should say I have no stakes in the company.
I see. It's not really an SPM. It's the raw material for your body.
It jumps. No, it's actually a step above that. It jumps starts, so it skips part of the steps because it's formulated in a way to provide and improve the conversion to SPMs over sort of general omega-3s, I would say. So that's the advantage. Now, in terms of providing SPMs, that's sort of the clinical goal of
I see where you're going. You take, so I think the SPM comes from the DHA portion of the omega-3s. So what your group hopefully will end up doing is taking that and putting it through the enzymatic conversion where you actually have SPMs in a capsule to give somebody.
Correct. So it's not just DHA. I should add it's DHA and 3-DPA, so the omega-3 version of the docosahexaenoic acid and icosapentaenoic acid. All three fatty acids contribute to SPMs. Right. So what we're actually interested in, and the aim of where we're, from a drug perspective, I'd say the supplementation perspective is very important. And I think it's critical because there is evidence in the literature that providing omega-3, including recently in a very big trial in cardiovascular disease, where people were given a refined form of EPA in the REDUCE-IT trial, there was clear evidence for benefit in reducing cardiovascular events, severe cardiovascular events. Mm-hmm. So I'm not saying that the supplementation point is not important. I think there is evidence for it, and it is an avenue that we're also exploring. But the other side, I think that in those conditions where the enzymes become disrupted, and we've shown this, that for example, in some patients, in specific cohorts of patients with vascular inflammation, the enzyme activity is lost. So in that case, you need to go with the mediator, the molecule itself, to reset the system.
Right.
And so in that case, the way we're exploring it is to actually produce these using a total organic synthetic approach. So you get a refined molecule that has the biology of interest that we can target to help reset the system. And then, to press that reset button, really. And then once you've reset the system, you can go to a supplementation approach.
So talk about the enzyme for a second. Is this enzyme something of, say, the cytochrome P450 cascade in the liver? And what is the enzyme?
So there's multiple enzymes, and we've characterized, and these enzymes have been studied for many decades. Some of these, but they're still unknowns. So the ones that we know of are the lipoxygenase, the family, the cyclooxygenase family, in particular cyclooxygenase 2, and there is a contribution of the cytochrome P450 enzymes too, as well as the soluble epoxide hydrolase enzymes and the epoxide hydrolases. So there is multiple families, and then there's also glutathionase transferases and gamma glutamate transpeptidase and dipeptidase. So there's multiple enzymes. The family of molecules contains over 70 members, and some of the pathways overlap, and some of the pathways are separate. And the interesting thing is that the pathways and the enzymes are shared with those molecules that are classically thought to be inflammatory, the prostaglandins and the leukotrienes.
Right.
So, and we think that this is also why, in some aspects, especially the chronic administration of inhibitors to these enzymes can have detrimental effects, because maybe in acute settings where you have overproduction of, say, prostaglandins, you will break that production and it's protective. But over a longer period of time, then you're influencing the pro-resolving mediator pathways, and that starts to lead to side effects.
This is fascinating stuff. This is exciting to me, because if you can actually produce the final end result molecule that's missing, and you don't require enzymatic conversions or anything else that could go awry from the precursor to the final product, then you can treat rheumatoid arthritis and a bunch of other pro-inflammatory diseases, I'm sure, with something that is more holistic and natural for the body.
Correct. And that's the aim. So our aim, or at least the hypothesis that we're working with, is that if we can engage protective pathways that are geared towards doing specifically that, terminating inflammation, repairing damaged tissues. We've shown in the past that, for example, some of these pathways can regenerate bone in the periodontium, even in large animals, in mini pigs, for example.
Mm-hmm.
So if you can engage these pathways without disrupting, and this is the other interesting aspect of the biology, that they do not suppress the immune system. So we've shown that they're not only protective in say, rheumatoid arthritis, but also in infection. So it suggests that you're not interfering with the ability of the body to deal with another inflammatory insult or an infection if it arises. You're just reprogramming it. You're just gearing it better so that it doesn't overshoot. And this, we think, will help a lot in minimizing side effects from treatments too.
Very exciting stuff. And I love it because it's like I said, it's a natural, it's something that we should be getting from our diet. As you said, by the time the train has run away, the brakes don't work anymore. So you can actually kind of go in there and rescue the person with this. That would be wonderful. Listen, this is fascinating stuff. I hope as you get closer, you'll agree to come back on the show.
Absolutely. It's been wonderful.
Yeah, this is really good. Dr. Dalli, thank you so much for being here today.
Thank you very much, Carl. Thanks. Bye-bye.
And we're going to actually pull the plug on the show. We're all done. We hope you enjoyed it. We hope you got some good information. Please share the show, because the gut, autoimmunity, rheumatoid arthritis, this is all great stuff. You can help a lot of people if you share this show. Please share the show. We'll see you tomorrow with more Superhuman Radio. Talk to you then.